Thanks again, everyone, for joining us on this November morning. Dr. Daniel Rath is joining us from NRDC. He's going to provide some scientific expertise talking about formation, storage, organic carbon in soils, megagenomes, and long-term ag experiments. Daniel studied how land management, microbes, nutrients, and soil carbons interact over decades as part of his graduate work, and has worked with agribusiness and NGOs in the United States, India, and home country of Belize as a farmer, soil scientist, and educator. Also has a bachelor degree in biology from Carleton College, a master's in soil science from the University of Edinburgh, and a PhD from the University of California, Davis. He's currently located in Washington, D.C. Thank you for joining us, Daniel. We're really excited. I'm gonna let you kick it off.
All right, sounds great. Thank you very much, Christine, and thank you everybody at the Michael Fields Institute for inviting me to give this talk. I'm gonna just share my screen and make sure this works. All right, can you see my slides? Awesome. Right, so yeah, hi, my name's Daniel Rath. As Christine mentioned, I'm a PhD graduate from University of California, Davis. I worked on soil microbial communities and soil, microbiology in long-term experiments. I worked with Nicole. I'm pretty confident in saying I probably would not have finished my PhD were it not for Nicole's assistance. So shout out to Nicole. And today I'm going to give you a little talk about soil health, but not just about soil health, but about the microbial processes that underlie soil health and how some of the things that we think about, or you may think about when you think about soil health, are actually driven by soil biology. There is time set aside for questions at the end, but this is, I kind of envision it as a little bit more freeform. So if anybody has questions or like burning thoughts that they'd like to bring up, feel free to raise your hand or feel free to interrupt me. I practice it enough times that I should be able to get back on track if you jump in. So let's make sure that I can, okay. So start off, if we're going to have a conversation about soil health, it's useful to start from a place of commonality. So the definition of soil health, as you would find on the NRCS website, is the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. Now, this is a great definition because it encapsulates several of the very important concepts that underlie soil health. It has soil as a living ecosystem, soil as something that functions, that does stuff, and then the fact that soil not only supports itself, but supports multiple other ecosystems that are literally built on its back. However, I'm a very visual learner, and so I like to draw diagrams and draw lines and draw pictures, and so I'm going to try to describe soil health using a metaphor that we should probably all be very familiar with, which is human health. So for humans to be healthy, there are certain functions that we need to carry out. We need to be able to digest food. We need to be able to circulate blood through our body. We need to be able to breathe in oxygen and breathe out carbon dioxide. In much the same way, because the soil is a living ecosystem, it needs to carry out certain functions in order to be healthy. So in this case, however, you're not eating like a hamburger. The soil is eating organic materials, so residues such as leaves and roots. It's circulating, but it's not circulating blood. It's circulating water, which has dissolved nutrients and may have microbes in it. And it's breathing. So the soil releases many different types of gases: methane, nitrous oxide, carbon dioxide. But in much the same way that we need to do all of these functions in order to live and be healthy, the soil needs to carry out all of these functions. And so when you're thinking about the benefits of a healthy soil, when you're thinking about what describes a healthy soil, I'd love to maybe get a couple pieces of input from people. When you think of a healthy soil, what is the first thing that comes to mind? Or none of you have ever heard of the concept of healthy soil, in which case this will be a very different presentation.
I honestly just think of that flat black dirt, which I grew up on and now no longer live and farm on.
That's a great definition. It's like you look at it and your senses can tell you it's this like black, you see it rich, it's crumbly. You see that probably if it's black, it means that it has a lot of organic matter. That's like a really good description of a healthy soil. Like that's a really good indicator. Anybody else have a thought?
Daniel Dagney says good structure in the chat, and Catherine says chocolate brown aggregates.
Man, aggregates are my favorite. So yeah, aggregates are another really good one, right? So you have really, when you think of a healthy soil, you think of improved soil structure, you think of the ability of water to infiltrate. And I just threw a couple other things up there, probably improve nutrient availability, reduce disease incidence, high levels of biodiversity. These are all things that we think about, like the indicators that we think of when we're thinking of a healthy soil. And healthy soils are really important. There are a lot of benefits to having them. We rely on soils for food production. Soils are one of the greatest reservoirs of biodiversity on the planet. They are also very key in our efforts to adapt to and combat and mitigate climate change. And most importantly, especially because I went to school in California, they really help us to store and filter water. There's also discussion that there should probably be a sixth benefit added to this chart, which is that healthy soils can help improve the health of the people and the communities that depend upon them. But there's still a lot of research that needs to happen before we can draw a very strong link there. It makes sort of intuitive sense, but it's because of the methods that we have to understand how soil microbes interact with human microbes, we're not quite able to draw a strong link yet. So healthy soils carry out a number of functions. Healthy soils are very important. We have a rough idea of what a healthy soil looks like, at least this intuitive understanding. How do soils do these things? How are soils able to provide these benefits? And really, many of the benefits that the soil provides are driven by the soil biological community. And so it's this incredible diversity of form and function that exists sort of beneath our feet every day, but we're unable to see or often interact with directly. But in a very real way, the processes that this invisible majority carries out are what our lives and our communities depend on. To give you an idea of just how small these things are, I'm going to do a little exercise in scale. So if you take a microbe, which is one of the smaller forms that soil biology comes in, 5 microns, that's very, very small, and you equate that to the size of a person, then a silt and clay particle, which is about as small as a soil mineral particle is going to get, 50 microns, again, still too small to even see, is roughly the equivalent of a semi-trailer to us. A soil microaggregate, which is the smallest unit of soil structure that we understand, and if it was on your finger, it would be smaller than a grain of sand, is roughly the equivalent of the Statue of Liberty to a soil microbe. And a clump of soil, which is the scale that we are able to interact with, you walk outside and you pick up a clump of soil from the ground, maybe about 2 inches big, so not that big, is the equivalent of 2 Mount Everests on top of each other. So it's about 11 miles to a microbe. So you can see how within a tiny piece of soil, you can have space for a huge variety of different types of organisms, different types of microbes, different types of nematodes and earthworms, and a lot of space for different types of functions that they're able to carry out. Because I really like sort of visual representations. There are a couple artist renditions of soil biological residents that I'd like to show you. This is a drawing of a nematode. Nematodes are super important. They're kind of like the T-Rexes of the soil, I guess, if you're putting it in above ground ecosystem terms. They're often carnivores. They'll consume smaller organisms, but they're really important when it comes to both plant health and ensuring that there's nutrient cycling in the microbial community. Here we have difflugia, which they look like snails, but basically they're amoeba that use organic glues to stick whatever they find together and construct little suits of armor around themselves. You have testate amoeba that are able to basically grow suits of armor around themselves. These are like biological, like basically overlapping plates, and they sort of stick their heads out to feed. And all of these incredibly diverse and very tiny organisms are intimately associated with plant roots. Every point of light on this image is a microbe. And this image is of a plant root that is like, on the tip of your finger, it would like, you would barely be able to see it. So even on the smallest, tiniest, most sort of almost insignificant looking pieces of like plant material, there is an incredible community of soil biological organisms that are working together in this gigantic living network. So when I'm talking about soil biology, it's not just the tiny microscopic like organisms, it's plant roots as well, and it's earthworms, and it's animals such as like voles and ground squirrels. You're talking about mites, you're talking ground-dwelling birds, you have like owls. All of these organisms are related and part of the healthy soil network that allows the soil to carry out these functions. The functions that we highlighted above, improve soil structure, infiltration, supporting food production. All right, so I'll take a quick second right there if anybody has any questions or anything that popped to mind while I was talking about that. And we're moving. Oh, go ahead.
Can you go back to the last slide, please? Can you just define what these trophic levels are?
Oh, yeah, absolutely. So trophic levels, think of it, it's, they say it's a dog eat dog world out there. And so in that case, these are the different levels of dogs. So when we think of what we eat, the first trophic level are organisms that are able to produce food via photosynthesis. And this is mostly plants. Every successive trophic level eats the one before it. So the second trophic level would be organisms that are able to break down plant materials. And this can include fungi, this can include, sort of whatever organ, like cows technically would be on the second trophic level because they consume plants. And then every successive trophic level going up consumes the one before it. So at the third trophic level, if you're going to think about that in terms of above ground ecosystems, it would be, I guess, a wolf that consumes a cow, and then so on and so forth. Humans would be kind of right at the top of the trophic levels because we eat everything, and not much eats us.
Thank you.
Yeah. Okay. So now that I've kind of like given a very broad overview of like soil health, why it's important, and just how amazing soil biology is, and how everybody should do a degree in soil microbiology, I'm going to talk a little bit about some specific examples that describing just how soil biology drives soil health. And I'm going to use two examples that I think came up when we were like listing what makes a healthy soil. Number one, how does soil biology help to both create and improve soil structure? And how does soil biology help us to store and cycle the carbon and nutrients that are stored in soil organic matter? And I'm going to start off with healthy soil structure, or soil structure. So when you think about soil structure, that's often the thing that comes to mind first when people are thinking about a healthy soil. You can walk out and you pick up a nice, beautiful aggregate that like holds together when you pick it up from the ground. And that is soil structure, but it's not the only part of soil structure. So the big piece of soil that you pick up holds together because the particles in that clump of soil stick to each other more tightly than they stick to whatever is around it. Now, if you have a lot of those aggregates or a lot of those big clumps of soil together, they're not going to fit together perfectly. And so just as important as these clumps of soil are the empty spaces between them. These empty spaces, because these aggregates don't fit together perfectly, are what create what's known as a soil pore network. And this is, in a word, basically the same as a circulatory system within our own bodies, right? Instead of carrying blood with dissolved nutrients and white blood cells, the soil pore network carries water with dissolved nutrients and microbial cells. In a very real way, is how things get around in the soil. They're also equivalent to like highways, And so when you have a compacted soil, what happens is that either through tillage, which may have destroyed those like aggregates, and like if you think about it, if things are smaller, they can fit together more tightly. And so that means that there's not as much space in between them. And so very often that's what happens when you have tillage, is that you break these soil clumps up into much smaller clumps. They're able to fit together and you don't have as much space for things to move between them. In this way, same thing with compaction. You compress it and you get sort of squish them together and you remove the spaces between these clumps of soil. It can, and if you're using that same metaphor, it's kind of like a heart attack when your arteries narrow and, water and air are no longer able to move. However, it's also has an issue for like plant growth, because, it's not just water and dissolved nutrients that are moving through these holes. It's plant roots that are trying to grow. And so if you have very thick compacted soil that does not have a lot of space for things to grow, to move through, you're not going to get good plant root growth. You really want that like nice porous structure. And then when you have very compacted soil, you end up with something like this, where you have, poor infiltration, water's not able to move, you'll get ponding, and that can sometimes either lead to reduced plant growth or increased disease incidence. So these clumps of soil are very important. They're what create soil structure and what create that pore network. How do they come about? It's not just, a large part of it is interactions between soil minerals that are able to hold together, but that represents overall, especially in the Midwest, a small fraction of like the total soil aggregates. Really, how these aggregates or how these clumps are formed is through the action of microbes and fungi. So you'll have soil particles that are sticking there, and microbes are able to produce these microbial glues that glue soil particles together. And then both fungal hyphae and roots will come through and form like this little mesh bag or net that like goes over these aggregates and really holds them together. And that's what you can see right here, is this like dense net on the outside that's like holding all of these soil particles together. The combination of these microbial glues and the nets from the fungal hyphae are really what allows soil structure to reform after it's destroyed. So when we have, again, when you have a tillage event coming through, if you'll notice, if you leave it for a while, that those big clumps will reform. That's just the action of microbes. The reason that they do this is that it's like building a house for themselves, right? Microbes will often associate really tightly with these soil particles because a lot of times there's like organic carbon on these soil particles, which is what they consume. And so they're basically building their houses. It can also help, these microbial glues that they produce can also help defend them against like, heavy metals, predators, a whole bunch of other things. So to summarize, healthy soils need good infiltration of both water and air. These soil pores are very important for the movement of that water and air. These soil pores are the empty spaces between soil aggregates, and these soil aggregates are created largely through soil biological processes. The biology of the soil is what creates the structure. Any questions before I keep going on? Anything that like people have burning curiosity about? Okay. I will
wait.
just keep. Oh, go
I
ahead.
have a
Sorry.
quick question.
Yeah.
Are these glues, are they costing the microbes carbon or nitrogen? What does it cost the microbes to make these glues metabolically?
Yeah. So this is really interesting. So the microbial glues are basically the microbial response to too much carbon. So if you think about, there's a lot of focus on carbon to nitrogen ratios, right? We want to make sure that there's enough nitrogen to the amount of carbon in the soil so that these microbes can, break that down, they can incorporate it into their biomass. If you have not enough nitrogen, but you have a lot of carbon, the microbes aren't just going to leave it there, they're going to find some way to make use of it. One of the ways that they do this is to convert that carbon into these glues. And so they basically will stick all these little carbon molecules together, and it'll be very, excuse me, they'll be very sticky. And they use that as basically scaffolding to build their houses. So it's really just a response to like high levels of carbon available in the soil, and microbes being, I'm not going to just leave it there, I'm going to find a way to make use of it. So, okay, awesome. All right, so the next one, which is healthy soil and organic matter, how microbes interact with organic matter, is one that has gotten a lot of attention recently because there's this big focus on organic carbon in soils and how in order to prevent climate change, we need to store organic carbon in soils, and your soil is healthy if you have high levels of organic carbon in soils. And while organic carbon is a really good indicator of soil health because it is an energy source, it is what powers the soil microbial functions, it's not the only thing, right? You don't just, if you just focus on soil carbon, you miss a large piece of the picture. And that's because organic matter, what we think of, that black rich color when you dig up a handful of soil, is not just carbon, it's a whole bunch of other nutrients. It's basically made of broken down plant, animal, and microbial remnants, and it contains nitrogen and phosphorus, potassium, and sulfur. If your plants are not getting nutrients from things that you add to the soil, if you're not, they are getting it from soil organic matter. So, especially elements like phosphorus that are kind of hard for plants to access when they're in their mineral forms. And again, it's an energy source for soil biology. So that carbon that you see in soil organic matter really represents the distilled energy from the sun that has gone through photosynthesis and has been trapped in the soil. So it basically is like a battery that powers the little Energizer bunny that are all of these microbial communities that we have. And organic carbon and nutrients come from many different sources. So above ground biomass is really important, and that's crop residue. Amendments such as manure and compost are also really important. But what's extremely important and often overlooked is below ground biomass and root exudates. So especially for the living part of the soil, the microbes that already exist below ground, roots represent like a very significant input of both carbon and other nutrients into that below ground ecosystem. That's just the roots themselves, like the actual biomass, but roots also exude a lot of carbon. And this is because roots are trying to attract microbes in this symbiotic relationship with the plant roots, because those microbes make more nutrients available to them. So when you're thinking about these source of organic carbon, roots and root exudates represent like a really big component, especially for these below ground ecosystems. And just kind of to highlight that it is not just carbon. A lot of the carbon input to the soil comes in the form of cellulose, which is the raw material that makes up, a lot of these leaves and stems, as well as lignin, which is a very complex molecule. And as you can see in the molecule on the bottom is the chemical formula for cellulose, and you can see there's nitrogen in there, there's oxygen in there, there's hydrogen, there's a lot of things that can be liberated by the microbial community. The molecule on the right is lignin. And really, I just put this up here to show that lignin is an extremely complex molecule that does not have a set structure. And so it's very difficult to break down, which is why if you bury a stick and a leaf in the soil, you dig it up a couple months later, what will often be left, some parts of the stick might still be left, and you might also see that skeleton of the leaf left. Those parts that are left behind are made out of lignin, which are very difficult to decompose. So if you're how do you go from what we recognize as plant residues to this sort of rich black organic material? it's the microbes. So the microbes and the soil biology are what mediate this transformation into what we think of as organic matter. I'm going, again, I like photos, so I'm gonna try and represent this in a series of diagrams using my excellent artistic skills. These are microbes, but you can tell because they have teeth. But this is an example of a below ground ecosystem that has a cover crop growing on it. And there are microbial communities that are inhabiting these roots. If we come through and we plow that cover crop in, all of a sudden you have all this organic material available to microbes in the soil that's there to be broken down. Now, microbes are going to multiply and they're going to start consuming this organic matter. And what they're going to do is they're going to take it in and incorporate it into their cells. So they're going to use that carbon and that nitrogen, that phosphorus, that sulfur, whatever is in that organic material to build structures inside themselves in much the same way that the food and nutrients that we eat help us to grow. Then, when those microbes die, their cells disintegrate. And that carbon and those nutrients are released back into the soil in the form of cellular structures and proteins. These cellular structures and proteins are much easier to break down than the very hard, like lignin and cellulose molecules that require like specialized like individuals to break it down. And so that carbon and nutrients, those carbon and nutrients that are released from the soil microbial bodies that are dying are what can then be made available to plants. They can promote the formation of soil structure. They can contribute to CEC. And that organic material is what we think of when we make statements like organic material increases infiltration, organic material increases water holding capacity. Really what we're talking about are plant inputs that have been processed by the microbial community. And one of the reasons that we really have a big focus on fungi, I don't know if you've talked about soil health, there are often people that are very, have very active discussions around fungi. It's because fungi are basically the only ones that are able to break down this lignin. So again, lignin is a very complex molecule. And so what fungi do is they load up this like very complex cocktail of enzymes and just shoot it at the lignin, and they kind of hope that something will like make a crack that they can then exploit. So these fungi in a very real way are the gatekeepers governing how the energy trapped in a large part of this like plant biomass flows into the soil ecosystem. So again, just to summarize, healthy soils, they store carbon and nutrients in soil organic matter. It's not just carbon, it's nitrogen, it's phosphorus, it's all of that good stuff that we want to make available to plants. That soil organic matter is created from the breakdown of plant inputs by soil microbes. And these microbes, especially fungi, control, they mediate the transformation of these inputs by processing that carbon within themselves. So I've talked a lot about soil health, and I've talked a lot about some of the microbial processes underlying the benefits of soil health. Why do we want more organic matter? Why is more organic matter good? It's because it's a store of nutrients and energy from microbes. Why do we have soil structure? Why are microbes important in soil structure? Because they are the ones that create both structure and the pore network that allows water and nutrients to flow. If you want to improve soil health, what do you do? The basic principles, and this is sort of from the NRCS website, is that you want more living roots, because again, those living roots are what represent a huge input of both carbon and nutrients to the microbes. You want to keep the soil covered because it's very difficult for the soil to carry out functions if it is no longer there and has been eroded. You want to disturb it less because when you destroy soil structure, energy has to be spent in recreating that soil structure in order to like recreate that pore network. And we want to cultivate diversity, and that means both above ground and below ground diversity. If you have a wide variety of microbes that are able to carry out a wide variety of functions, you have the potential for greater resilience. You have the potential to respond better if you have like a major disturbance. And these are basically the principles of soil health: minimizing disturbance, maximizing biodiversity, maximizing soil cover, and maximizing living roots. We've talked about why soil health is important. We've talked about sort of what are the things you can do to like promote soil health. I'll talk a little bit about how we measure soil health and like what are some of the indicators that can be useful. One of the things that can be useful is using soil tests. So if it depends on what question you want to ask to begin with. Are you measuring soil health because you see a specific problem in your field? Maybe you see poor infiltration. Maybe you're seeing increased incidence of disease. Maybe your plants are not doing well because the pH is off. That informs the tests that you would do in order to in order to ascertain what the extent of the problem is and how to fix it. However, if you are measuring soil health to get an understanding of how your practices are improving the soil, you can use any one of these soil tests, or you can one of the methods that have been cited as like one of the best ways to understand soil health is just using your 5 senses when you go out onto the field. And that involves looking at the soil color, looking at aggregates. Do you have good soil structure? Do you see a lot of ponding? Do you see a lot of infiltration in particular parts of the field after it's rained? You can do these sort of formalized measurements, like such as aggregate stability, which is you put an aggregate in you submerge an aggregate and you see how long it takes for that aggregate to dissolve. And if it takes a long time to dissolve, that's a pretty good indication that you have good soil structure, because again, those microbial glues and microbial nets are holding it together. But really, it's not a substitute for your interaction with a particular plot of land. Being out there every day, interacting with it, you get a very innate understanding of how the soil is changing and potentially improving due to the practices you do over time. The NRCS has a really good sort of guideline to in-field soil health assessments, and I know that the folks at Michael Fields Institute have a really good guide to soil tests and what are like some of the how to interpret some of these tests, what are the underlying mechanisms behind them, and whether or not you should do them. So I'm not necessarily going to go into that. I think these guys have a pretty good are pretty good resources on that already. But I do want to highlight that soil health is a sort of it depends on your field. Every soil, if you want to increase soil health on your field, it's not just you just look at the list of soil health practices like cover cropping, you pop cover crops on there, okay, the soil is going to get healthier. Every soil health practice is dependent on the context of your field. It's dependent on your soil type. It's dependent on what you grow. It depends on your goals for that particular plot of land. What is the climate like? Do you get a huge amount of rainfall? What is the terrain like? Are you near like a sort of low-lying swampy area? All of these factors are going to influence what soil health looks like in a particular plot of land. If you want to compare your plot of land to some other indicator and see whether or not it's improved, really the best thing you can compare it to is that same plot of land like 3 years ago, or that same plot of land under a different set of practices. And this is why it's really important to test and interact with soil regularly, especially if you're thinking about soil health, is that seeing how that soil changes year to year, how it reacts to certain practices, can often give you a better indicator of whether soil health is improving or not, as opposed to sort of comparing it to like a random field somewhere else that may not have the same conditions and may not have the same constraints that this particular plot of land does. I've talked a lot about soil biology, and one question that I always get is, what about biological soil amendments? You want your microbes to be happy. I've just spent like a good hour talking about how soil microbes are like super important, and how how do we promote like their health and how all these functions that they carry out. So it's like, oh, shouldn't we be able to like put this thing on there that's like specifically formulated to target microbes? I cannot, like, you know, as a scientist, I haven't seen a lot of data about the outcomes of specific amendments. However, I do know that we don't know whether if you have these formulations that say that they add a beneficial microbe to the soil, it's not clear whether those microbes are able to survive in the soil for long periods of time after you add them. It is a fiercely competitive world down there. Like, there are these very tight, like, prey-predator interactions, these very tight competitive interactions may not have a lot of space for like new soil microbes to be introduced. It's also unclear whether even if you introduce like a specific organism to the soil, whether it's actually going to increase the soil's potential to carry out a process. So you might not even see like a strong effect. If you're like, I want to promote the health of the biological organisms in my soil, honestly, the best way to do it is to feed them and house them. And this just links back to the general principles of soil health. You want to put more carbon inputs into the soil, whatever form those carbon inputs take, it compost, cover crops, crop residue. That organic carbon and those organic nutrients are, again, the energy source for these microbes. You also want to reduce disturbance and erosion, because again, if these aggregates, if these soil structures are, for lack of a better term, microbial houses, destroying them means that they have to spend energy regenerating them. And so you want to be kind of careful about the timing and the amount of tillage you applied, and keep the soil covered to prevent erosion, whether that is in the form of cover crops, in the form of mulch, etc. All right, I have like 1 minute left according to my watch. It was 45 minutes. I'm going to do very quickly soil health and perennial and grain systems, but we do know that diversifying crops can improve root penetration, carbon and microbial biomass. We have a pretty good idea that perennial grasses and legumes in rotation can give a lot of different ecosystem services. Again, a large part of this is because roots play such an important role in the soil below ground community. You get improved nutrient water cycling efficiency, they enhance climate regulation, and they improve soil health. Because I'm a scientist, it wouldn't be right for me to go through this presentation. I have at least one graph. I will say it is not my graph. It's a graph from one of my collaborators, Kaylin Diederich at UC Davis, that was growing perennial wheat and perennial wheat, no-till wheat, and tilled wheat in the Russell Ranch experiment. And I do appreciate that I'm showing a graph from California to a bunch of farmers for the Midwest. I hope you'll forgive me for that. But I thought that this was a really good indicator of how different wheat systems can have an impact on soil carbon. So what we're measuring on this graph is mineralizable soil carbon. So that's soil carbon that, for lack of a better term, can easily be broken down by microbes. And what we see here is that the blue line, which is the Kernza, has higher levels of mineralizable carbon sort of throughout the profile than either the no-till or tilled wheat. And in large part, this is due to the fact that that perennial wheat, those roots exist for longer periods of time in the soil, more carbon inputs, but also more microbial cycling of nutrients. Okay, and with that, I invite any questions, or I know we have a little break and then time for discussion. So, please feel free to fire away.
Thank you so much. Yeah, and also don't rush through this part, because this is a group of grain farmers. So this is the information we're all pretty jazzed up to hear about. So folks, I know that's a lot of information. Feel free to stretch and do your thing. But Daniel, there was a question. Oh.
Go ahead. Yeah, go ahead.
There was a question in the chat that Tim just asked, but he said you kind of indirectly answered his question with one of your last few slides. But if you could maybe touch again on, are microbes created equal? Are more always better.
Hmm.
Microbe dealers feel like they're vitamin salesmen. What is quantitatively best for our soils?
Yeah. These are all very good questions to do PhDs on, honestly. So, are all microbes created equal? No, I would say that, as in, I'm going to go back to this slide on soil health, right? In that everything needs to be placed in context. There are soils, we have identified specific soil microbes that are better at cycling nitrogen, right? And that's great. However, if you have a field that like has a ton of nitrogen in it, like those microbes aren't necessarily going to be important. And so all microbes are not created equal, they perform different functions, but whether or not that function is important to you depends on the specific issues that that field is facing. When you think of these amendments as like if you're saying like are these amendments like vitamins, do you just like, are you able to just like boost the soil microbial community? These inputs happen over longer periods of time. So sure, if you have if this a jug of whatever you're adding, it holds like dissolved carbon and dissolved nutrients in a form that are easily broken down by microbes, and you add that super regularly over several years, yeah, there's a lot of potential that the microbes in that field will do better. However, it's not immediately clear that that's like a whole lot different from just like growing cover crops and putting those, like plowing those in, like the same principle. You're just adding carbon to the soil. And so it kind of depends on what form that like is sort of like best for, like what form that is most convenient for you, I guess.
I would add that
Mhm.
from a practical product testing standpoint, when Daniel and I were at UC Davis, there was a professor that got 50. Patrick Brown, Daniel, he had like 50 different products that he got from different biological formulation companies. And he tested them all according to their label of application. Of the 51 increased cash crop yields, maybe 2. So it's not that there aren't products out there that can make a difference, but they're few and far between. There's no independent verification process for testing these, so you have to rely solely on the company's claims. So I would say with these bugs in a jug, there are some that work, but it's very difficult to identify which ones those are. And there's a lot of people trying to make a lot of money and not offering very solid data at all. So if you're, yeah, I'll leave it there.
Yeah, I had a whole other presentation that I did on sort of like soil microbial tests, and like metagenomic analysis, which is like this really deep look at like the DNA of like organisms that are in the soil in order to try and describe just how things have changed over like decades. And we did it at the Russell Ranch experiment. And what we found was that if you are trying to like improve like the soil microbial community overall, we had 25 years of data from an organic system which had compost and cover crops versus a conventional system which had like mineral fertilizer and pesticides. And after 25 years, if you just looked at the overall soil microbial community, they weren't that different. And a large part of that is because soil microbial communities are strongly controlled by like soil type and climate. However, if you zoomed in and you asked very specific questions about, okay, well, I know overall it doesn't look very different, but like maybe if we're just talking about like the ability to break down carbon, is it different? And we did find that the organic system had a greater ability to break down carbon. And so when you're talking about sort of like these biological formulations too, like even the results are like it increases soil microbial communities, like it increases the amount of soil microbes. it's like, okay, increasing the amount of soil microbes is great, but it's not immediately clear that that's going to have strong changes in function or strong changes in the things that the soil is able to do. So, I can't see the chat until I stop sharing my screen. So if there are any questions, I can stop. Yeah.
Relay them back. Steny had a question, and thank you, Tim, for asking that. Steny had a question. I'm just going to read. In your example of microbes eating plant organic matter, you mentioned the cover crop was plowed, but then also talk about no-till benefits to soil structure. So in a no-till system, is it mainly the roots? And over time, the surface contact of the above ground vegetable breaks down, i.e. eaten, or is it worth plowing in green manure?
Yeah. So no-till is a really good example. So no-till originally, to my understanding, was created back in the 1950s as a way to reduce soil erosion in like the wake of the Dust Bowl. And so, but it just happens to have a lot of benefits because you're reducing the amount of disturbance that happens to the soil. And so with cover crops, if you're the cover crop breakdown from a purely scientific standpoint, basically the higher amount of surface area of your cover crop that is in contact with the soil, the faster it's going to be broken down, because that is more surface area for like microbes and fungi to act upon. Whether or not it's better to plow that green manure in or leave it on the soil, honestly, I'm not an agronomist, so I couldn't tell you. Perhaps Nicole could answer that one better. But broadly, if the residues are going to break down, they'll break down faster if they're plowed in. Whether or not that is worth the benefit, because you do get that benefit of soil cover if the residues are on top of the soil, there's potential for less erosion. But whether or not one of those balances out the other kind of depends on your situation. I will say that when you think of a disturbance, rain is actually a pretty good disturbance. If you do an aggregate stability test, you take an aggregate and you submerge it in water and you see how that aggregate dissolves. What you're doing there is simulating on a bigger scale what happens when it rains. Water hits the soil and those aggregates can fall apart, they can dissolve, and all those particles can move down and clog up the soil pore network. And so if you have cover on the soil, you can also potentially increase infiltration and reduce the mechanical impact of rain hitting the soil and potentially disrupting soil structure.
I would say in trials that have compared no-till to tilled termination of cover crops, they haven't found huge differences, at least in the short term. Over the long term with no-till, you do tend to get stratification of nutrients. So what I hear recommended increasingly frequently is intermittent or rotational tillage. So some guys think about that as like tilling every 5 years.
Yeah. And that is better with perennial crops. Or Katie asked, or strip tilling.
Yeah. Strip tilling's kind of the best of both worlds if you can do it. And some people keep strip tilling in the same zones and some people move over. So you have rotational strips that are tilled and not tilled. Do we want to read Dagny's question, Christine? That's a good one.
Yep. Right, coming up next. Can you comment on the different soil carbon pools and whether it actually makes sense for farmers or land managers to say we are sequestering carbon?
That is also a really good question. So if as a researcher, if I'm trying to research whether or not soil organic carbon is stored, there are a number of different pools that I'm going to look at, whether that's particulate organic matter, mineral associated organic matter. if my goal is to say, am I sequestering carbon depending on the amount of land you have and like the availability of these tests, there's a reason that most people will just do total carbon tests, because they're like relatively easier than doing, again, sort of like mineral associated carbon, which is better, like in itself is a like stronger indicator of whether that carbon is going to stick around for a long period of time. As a farmer, if you're trying to say that you are sequestering soil carbon, one of the best ways to do that is to probably track that carbon change over time in your soil. Total carbon, you can, it depends on like your initial carbon level as well, because you might not see big, if you have a huge, if you're like carbon percentages, I don't know, for California, 1% is huge, right? So I would imagine maybe like 5% or 10% is more relevant. But if you see a change of like 0.5% or 1% in those soils, it might be harder to observe that through like total carbon measurements. And so but that's sort of the standard that everybody wants if you're talking about sequestering carbon. If you're talking about potentially mineralizable carbon or POC, those aren't really clear indicators of soil carbon sequestration, because really what those are telling you are like what carbon is available for microbes to use as energy. And that's the dichotomy there, is that you want to increase soil organic carbon because it's a climate change like mitigation method, but it's also you want to use soil organic carbon because it's a great store of nutrients, it's a great store of energy for all of these other processes that we need to carry out. I don't know that if that was super helpful. I didn't give you I didn't just point to a indicator and say measure this, and that's the best way to say that you've increased soil organic carbon. But I know, Nicole, I don't know how available it, if I was doing it, I would do density fractionation and look at the soil carbon that is closely associated with minerals, because that's this carbon that's going to stick around for a long period of time. However, there's a reason that nobody does that, is because it is, excuse my French, let's just say very, very difficult to do. And it is not it is deeply unpleasant experience. So total carbon is often the best indicator for a reason.
I would say, in reviewing all the literature and the wealth of studies conducted, perennial grasses in particular, and these can be in a prairie or a native grassland or a grazed pasture context, it's really the only agricultural system that has been consistently shown to meaningfully sequester carbon, which means that those soils are growing in carbon over time. So they're a sink for atmospheric carbon. It is that reason that I work on Kernza, because it can be a solution for farmers to sequester carbon and produce grain. The economics of that system are still being worked out. In some cases, like what Daniel and I worked on in California, repeated compost and manure application did increase carbon. The trouble also with monitoring carbon in today's day and age in ag soils is they're often only doing it in the top foot of soil, with the rationale that that's the depth fraction of soil that farmers interact with and influence. The work that Daniel and I were doing was finding that there are carbon changes in the subsoil, even below the plow layer, that are influenced by what goes on at the top. And so some practices, some systems were resulting in more carbon gains in the soil profile than what we were seeing in the top, which means we were underestimating carbon gains. In some systems, the opposite was true. There were losses at layers deeper than the plow layer, which means by just looking at the plow layer, we were overestimating carbon changes. So it's good to know that practices if we want to track soil carbon, we should be doing it deeper, like 3 feet or more, 3 feet minimum. And that, I hate to say it, but well, I saw a presentation yesterday of 77 studies, like only looking at carbon change with cover crops. Only 22 of them found that cover crops increase soil carbon. So different practices work differently on different soils, but some have a better shot than others. Let's put it that way. I hope that's helpful for farmers thinking about carbon markets.
Oh, yeah. There's a lot to say about carbon markets. But it's, yeah. Anyway, that's not the question, so I'm not gonna ask about carbon markets.
And since you just touched on it a little bit, can you dive in a little bit deeper to the types of cover crops that may be better for soil health than others, or not necessarily?
Sure, it can. I'm not cover crop expert, but I will give you what I know. I know that very often, if there's so you want a good carbon to nitrogen ratio is often the point of cover crops, because if you grow something, there's like, a rye cover crop, at least in California, we do like a oat cover crop. That's a lot of carbon in that woody biomass. And so you want often a legume to balance that out. And so I know that cover crop mixes of, what did they have at Russell Ranchers? Oats, vetch, and like fava bean. But just the fact that you have that combination of like a high, like sort of carbon content cover crop combined with like a high nitrogen content cover crop. I know we've seen that particular cover crops, especially was it daikon radishes, I think can help loosen up soil compaction. So again, it depends on what your issue is. If your goal is to grow cover crops to like increase compaction, you're going to want cover crops that maybe have very robust root structures that are able to like pierce that like compacted region. And yeah, I mean, there's been a lot of research on cover crop like mixtures. Like, we talk about increasing above ground diversity in order to increase below ground diversity. And I read a couple of papers sort of before this talk on like what is the right like amount of species for cover crops, and it seemed like it like averaged out like 8 different species was when you saw like maximal biodiversity benefits. There was one mix that had like 24 different species, and they didn't really see like a strong effect. So you probably get like, in a way, diminishing returns depending on like the number of species that you add. Nicole, I don't know if you have any thoughts on like specific cover crops that are like good specific species.
Been a lot of research on this, and there's usually not a lot of separation in the data between cover crop types or mixes. I hear that a lot of farmers try different things and then they really settle on one they like. So I think experimentation on your farm with different cover crop mixes is good. We have pretty restricted options in the upper Midwest, particularly for legumes. There really aren't any legumes that have winter survival right now, besides perennial types like clovers. And that's kind of an investment to make, because if you just want to have a winter cover crop, then clovers take a while to establish, and then you're just going to terminate them, blah, blah, blah. There are some winter hardy varieties of winter pea that are supposed to come out in a year or two. And so I think that'll be huge if we can get pea into cereal rye. But really, the only practical option for people north of probably Chicago is cereal rye. But if you're south of there, you can do crimson clover, you can do other things. Vetch is an option, but it causes a lot of contamination in small grain crops. And as just a blanket recommendation, I advise people to stay away from it.
Yeah, you want to be kind of careful about cover crop recruitment in the next year. And so there's like active discussion about what is the best way to like terminate a cover crop, right? And I think one of the things that always stands out to me about like specifically no-till systems is that it's off, like you want, I guess crimping and rolling is like the main way. we used to mow and then incorporate. And then of course there's like the herbicide option, which is the impacts on soil biology are not like super excellent, but like that is sort of like a option that's used.
Great. Folks, we have a good amount of time to ask specific soil health questions, perhaps that you're experiencing on your farm, or dive in deeper to some of these topics that Danielle so eloquently explained for us. So don't be shy. This time is for you to make improvements where you hope to see them. So jump on in. Thank you. Michelle, go ahead.
So I thought maybe we were going to get into the decontamination portion of his first couple of slides. We talked about that, with the body can help to decontaminate itself, and the soil can. I was at a meeting, a field day for tillage a week and a half ago, and they actually were talking about how pesticides and herbicides and chemicals that are applied to conventional fields will stay indefinitely. It'll link to the organic matter and can stay in the soil profile indefinitely. I had not heard that before. The question originally was, how long will Roundup stay in your soil, glyphosate? And they said years, and then they got into the indefinitely part. So understanding organic matter in the limited sense that I do, I'm wondering if he can touch on if there's any way to actually decontaminate it, or if it's just encapsulated in organic matter and it can never leave.
Yeah. That's a really good question. So there's one sort of informal mantra that I repeat to myself, is that if you put it in the soil, something will find a way to eat it. Because the soil is like again, it's a very tough environment to exist in. And so like if there is a potential source of energy, some microbe will find a way to exploit that. Now, whether that is the kind of microbe you want in your soil and whether the byproducts are beneficial is another question. You are correct that like because of partitioning, which is that like some molecules don't like to be around water very much, and so they will like if your choice is water or organic matter, they'll like go stick into the organic matter, and so can like exist there for a longer period of time. I don't know about indefinitely, again, because something always finds a way to eat, like, you know, MTBE, like you can pour gasoline in the soil, and they're like sort of any kind of organic contaminant you find in the soil, something finds a way to break it down. Very often there are negative like byproducts, and the like microbes that the conditions that you want to break it down are not ideal, but something will find a way to break it down. However, that does not necessarily happen right away. And so you can have pesticides and herbicides like persisting in the soil, and especially persisting in high organic matter soils for extended periods of time. When it comes to getting rid of those, there's not like, as far as I know, there's not a really good way to like separate out sort of pesticides and other contaminants from like that. This is one of the reasons that like soil contamination, brownfield sites are so difficult and expensive to remediate, because very often the cost of fixing whatever was done to the soil is greater than the benefit you would get from it. And so often the solution is just to cart all the soil, like get a bulldozer, dig it all up, stick it somewhere, and then forget about it, right? Because it once it's in there, especially in the organic matter, it's very hard to partition back out. Hopefully that was good.
I just looked up a paper and it says the half-life for glyphosate is 200 days in the soil. So that would be like 2 years.
2 years or a year and a half.
Yeah.
Yeah. I mean, if like I said, something will find, if you put it in the soil, something will find a way to eat it.
I appreciate your answer. I do grow on an old conventional field that was conventionally farmed for years before I got it 15 years ago. And I guess in some ways I noticed what you say, because it has taken a good deal of time for it to grow the way I thought it could. You know what I mean? So I'm happy to hear that that's what happens, and you sort of ease my mind. Thank you.
This paper also does say that it does inhibit, glyphosate causes continuous negative effects on the soil ecosystem by inhibiting the growth of many indigenous microorganisms. And it does accumulate in the soil. So people are making repeated applications per year. So with that accumulation, which you're drawing down now,
Yeah.
Michelle, but yeah, I think maybe that's part of the reason why people say when you go organic, it takes 5 years. And there are other chemicals in there too that probably have longer half-lives.
Speaking on the organic transition period, there's also really interesting work on that from like a purely microbe point of view, which again, I'm biased, that's all I think about. But when you have an ecological system, a lot like as we do in the soil, the things that you do to it, the disturbances that we use, and basically everything that we do to agricultural soils can technically be termed a disturbance. The microbial community learns to adapt to that. So they adapt to a particular level of disturbance. If you are continually adding nitrogen, they adapt to no longer sort of release as much nitrogen from organic matter because they just don't need to. They want to have a more efficient use of energy. And so in some ways, the organic transition period is the soil adapting to this new form of disturbance. It's like more organic carbon. They need to release more nitrogen from organic matter. There's less disturbance, and so they don't need to focus as much. Like often there's less disturbance, so they maybe don't need to focus as much on creating soil structure. And like that period, like you said, that transition period, about 5 years is how long it takes for them to adapt.
Thank you. Folks, any other questions for Daniel? Soil issues you may be experiencing right now that you'd like to?
I'll say this is a great document. I think it's if you have worked on a farm before, nothing in there, I guess, is going to be surprising. You like go, you pick up the soil, you like interact with it, you look at it. I think when it comes to actual soil tests, again, you can measure a lot of different, you can measure mineralizable nitrogen, you can like kind of estimate the rates at which organic material is released from the soil. And that's super helpful if you want to get like sort of a snapshot. But I think to what Nicole was saying as well, you want to have a good idea of how your farm is in space and time. So in space, we mean by you want to look at how things change when you're going deeper into the soil. And with time, it's, you know, a measurement at one time point has limited utility. A measurement at multiple time points and understanding how things have changed over time really increases the benefit that you might get out of these soil tests, because you can then have a good idea of how your soil has responded to the management.
Thank you so much, Daniel. I'm gonna offer folks a last call for questions. I mean, I don't want to take space here, but I know they exist. They're percolating. Jump in, friends.
Daniel, we're compiling this soil testing info sheet to hand to folks. And Dagny asked the question that I was going to ask you to comment on, but Dagny asked, can you talk about some of the newer soil tests floating out there, like the Haney test, compared to just like routine soil tests that we recommend that everybody use?
Yeah. yeah, so the Haney tests, if I can remember exactly what is in contained in that, but it's like you look at soil aggregate stability, you look at soil pH, you look at soil organic matter, and what it does is you do sort of all of these things and you try and combine them into one indicator that gives you a good idea of whether your soil is healthy or not. And there's a couple other indicators like soil enzyme activity that are also useful. It's not, I would say, when I think about these, there are two contexts that I can think about these soil tests. Number one is I think about them in terms of gathering data in order to like form a hypothesis and in order to like explain like a process, and I write a paper about it. Then I think about the soil tests that I rely upon to like inform management and like what are the best indicators that will like tell me like what it is you need to do on the field. And I think there's one of the issues is that a lot of the newer soil tests are like more sort of, for lack of a better word, high-tech soil tests don't have a really strong link to management. And so like one of the newest soil tests is you take your soil and you get it microbially analyzed, and you'll have these companies that send you back this really beautiful looking report with like, here's the potential for all of these things that are happening in your soil. I have worked with the data that goes into those reports, and I will say that the level of uncertainty associated with those results is so high that it's almost like a sort of educated guess, right? And so there, in my estimate, if you're looking for soil tests that are like intended to drive management, very often they are the tests that have been around for a long time. I think specifically to like the Haney soil test and some of these newer soil health tests, they measure, they're a way of formalizing the like what you kind of know, or the interactions that you might have with the soil if you are going out, if you're able to like go out into the field. So yeah, I mean, aggregate stability, available nitrogen, organic matter, these are all way more helpful because you can kind of, there's a clear idea of how these measurements link back to like healthy functioning of soil. And so it's, yeah, I will say that I've interacted a lot more with the Cornell soil health test. And that one I think was quite nice because what they do is they give you an idea of the range that your field falls into. And so like, it's like, if you can have a number associated with a measurement, but it's kind of useless if you don't have something to like compare it to. And so I remember that they give you like a sort of a scale of like whether or not this is working. And at the end, you can have a soil that is healthy in particular areas. You can have really good nitrogen availability. You can have really good mineralizable carbon, but your penetration is not so good. So like, your soil is fairly compacted, or your organic matter might not be as high. And so, yeah, I would say that that's sort of my input on those.
I feel like this is kind of what you talked about earlier, where it's not necessarily the one test in 1 year, it's the change over time, and it's what the indicators that you're interested in measuring over time. And maybe the one thing in like a soil health analysis test that's actionable, might be looking at like the organic carbon and nitrogen ratios, and then developing a cover crop mix that is heavier in nitrogen or heavier in carbon based on where that ratio is. Yeah. If you wanted to increase your microbial activity, which is something that is also measured there. Yeah. But it's tricky because it's like these tests you can't just do once and then say like have actions all the time based on just one test. It's the change over time that you're looking for. Yeah, and that's you want to be really careful if it's just you have one test and it's like, okay, you need to hit this is the only test you need to do, and it will tell you everything you need to know. It always depends on what is the question that you want to ask, right? And so
like you can, again, it's hard. I always have found it very hard to talk about these things in abstract, right? It's like, is this soil health test good? It might be good. Like, what's your problem, right? Do you have bad aggregate? Like, are you worried about your aggregation? Like, sure, then this will tell you what you want to know. But is it like, why do I have like particular like disease occur? I have like a lot of disease occurrence. Like, will that help? Like, probably not, right? Because it's not going to tell you much about disease. So it really depends on the context.
Dagny, do these tests provide agronomically actionable information? I would not order the Haney soil test. It measures indicators that are very fluxy. So like with nitrate, you can get a nitrate test and it's not that helpful really, because it's constantly changing. It's doing this. So the things in Haney are things that are also doing that all the time. And so when something's really fluxy, you can't identify the root cause of why it might be high or low, because there's too much going on. So I'm not a fan of Haney at all. I agree with Daniel. The Cornell one is better. It measures too much stuff and it's very confusing. I think Hannah and I should come up with, I need to think about it a little bit more, but we can come up with something of more stable measures that can be more helpful. The problem is I don't think that my opinion of what is helpful is in a nice named package, but we'll do some digging and try to make some recommendations. But I agree about getting egg stability tests. Particularly, I find it helpful on different fields that have different histories. And then you can get kind of a feel yourself of how things compare on your farm, depending on what you did. And if you see one field getting better and you see a value ticking up, then you can, and it's not in the other fields, then you can kind of think, well, maybe that's because I've been cover cropping that one. But I want to get to Michelle's question.
Hi, I just have a simple question because I plant winter killed cover crops all the time. Because I'm planting vegetables the next day or the next spring, and I wonder if there's any value to that whatsoever after listening. So let me know. You know, oats and peas, or I put soybeans in for potatoes. I don't know. Is there any value in a cover crop that doesn't make it to the winter?
I mean, I would say, yeah, I mean, you have living roots in the soil, right? So like I would say yes. So you grow a cover crop, like you have something growing, you have root growth, you have that carbon input going into the soil, you have those roots creating channels that can, like, again, can potentially contribute to the pore network. You're potentially cultivating diversity. And so is it beneficial to the soil? Yes, I would say yes. Is it cost effective in terms of like the amount of effort that you spend planting it? I can't answer that. That's sort of something that only you can answer. But yeah, cover crops are, I like, anytime you don't want to see bare soil. Anytime you have plants growing in the soil or you have living roots in there, you are contributing to soil carbon and you are like contributing to soil microbial community.
Michelle, Tim also talked about weed suppression with cover crops. So
Yeah.
Awesome. We've got space for one more question, and then we'll let everyone carry on with their fabulous Thursday. So speak now or forever hold your peace. Or Daniel, can we share your contact information with our farmers? Thank you.
Absolutely. Yeah. I will say that I hope that this has at least given you some idea of, when we talk about, like, we say aggregate stability is important, but this, like, what I was trying to get at is, why is aggregate stability important? Why do we talk about microbes? Why do we care about organic matter and all of these things? And I think that when it comes down to specific management practices, sort of what I've seen time and again, the best scientific questions that I've ever encountered are generated from a 10-minute conversation with a farmer, right? Where it's like, I am not the one that has spent time on a particular plot of land. I have spent a lot of time studying these like processes in abstract, but very often the observations and the questions that are generated and that are most helpful come from the people who have spent the most amount of time actually interacting and like working with the soil. And so that's the part of the reason that I'm saying, like, look at how these things change over time, is that, you know, each of your farms is like a tiny experiment and you guys are the ones running it. And so you are the one that are best informed to like interpret those results.
Yeah. Wes from Granner Farm made a really great point earlier this spring, like every season is just one new experiment, and that's really, it can be intimidating, but it can also be really powerful. So dive into that and see what makes your fields thrive. Well, I'm going to end the recording, and thank you so much, Daniel, for your time and expertise and enthusiasm for this topic.